what structure of ovule. Already I have studied about the structure of ovule. Ovule is covered by one or two layer structure called integument. Integument is the white gap at the top called microphyll. Opposite to the microphyll is the chalaza. Then the integument encloses a nuclear tissue called nucellus. Homogeneous mass of cells called nucellus. One of the cells in the nucellus functions as megascope mother cell. It divides by meiosis to form 4 haploid megaspores. Only one megaspore becomes functional. 3 megaspores degenerate. That megaspore develops into functional megaspore develops into female gametophyte or embryo sac. Now the development of embryo sac already 5 more questions that one already I taught you. Now in that embryo sac, what are the structures you get? Egg and synergids, collectively called egg apparatus. So egg apparatus found close to the microphyll end. So one egg on either side, 2 synergids. There will be secondary nucleus. Said diploid one or it differs. Then opposite to it 3 antipodals. Antipodals are haploid. Egg haploid, synergids haploid. Now, this is what structure of ovule. Normally, what happens to after fertilization? After fertilization, ovule develops into seed. The fertilized ovule is called seed. Now, the fusion between the male and female, already I just told you last class. Amphimixis, fusion between the male and female gametes, female nuclei is called amphimixis. But apomixis, reproduction. But normally this is sexual reproduction. So when reproduction does not involve the union of male and female gametes, is called apomixis. That's what called reproduction does not involve the union of male and female gametes, is called apomixis. This term was introduced by Winkler. Mageshwari classified apomixis into 2 types: recurrent apomixis, non-recurrent apomixis. So recurrent apomixis you get various types: vegetative reproduction, then agamospory. Agamospory, adjunctive embryo, then diplospory, apospory. Then apart from that, I didn't take you that one non-recurrent apomixis. See, in recurrent apomixis, in the sense the young ones develops the young one develops. So from diploid condition, but whereas non-recurrent apomixis in the sense haploid one. Haploid embryo sac formed after meiosis develops into an embryo. This is what haploid, very important. Haploid embryo sac developed after meiosis develops into an embryo. But this embryo is a haploid one. That is why it is non-recurrent apomixis. So next one about polyembryony. Normally, generally, what happens to the ovule after fertilization? Ovule develops into seed. After fertilization, ovule develops into seed. So monocot seed, dicot seed. This was seen having 2 cotyledons. This part is a planule. This one is a radicle. So normally, which one develops into embryo? The fertilized egg develops into, the fertilized egg is called zygote. Zygote develops into embryo, the embryo from which you get all these things. So this is what normal seed. So this is what normal seed development. But only one embryo develops from one ovule, only one embryo. One ovule, only one embryo. From single ovule, single seed. Single ovule, single seed. Single embryo, single seed. Then one embryo develops from there. So this is what generally happening. But sometimes what happens here, the development of more than one embryo in a seed. Development of more than one embryo in a seed, that's what called polyembryony. See, polyembryony is a condition where more than one embryo develops in a seed. Normally, generally, only one embryo develops in a seed. But sometimes what happens is many embryos develop. So now, there are four types of polyembryony. The first one, cleavage polyembryony. So what is cleavage polyembryony? Zygote. Zygote divides into embryo. One embryonic cell, two daughter cells, and then two daughter cells separated into two embryo, different embryo. So this is just one example given to you. Okay, zygote develops into two cells, and then such two cells develop into embryo. This is what cleavage polyembryony. Division, cleavage polyembryony. So cleavage polyembryony in the sense only one embryo develops from that. But sometimes what happens, due to separation of cells, many embryos are developed. So that kind of polyembryony is called cleavage polyembryony. So, occurrence of more than one embryo in a seed is called polyembryony, first reported by Anton von Leeuwenhoek in 1719 in certain oranges. See, based on the origin, polyembryony is four types. There are four types of polyembryony. Cleavage polyembryony, as I told you already, cleavage means division of zygote into two cells and many cells and then to give one embryo. But here what happens here? Such cells separated, they may give rise to two or more embryos. That's what called cleavage polyembryony. Example, orchids. Orchids belong to Orchidaceae, ornamental plants. Then formation of embryo by the cells other than the egg and synergids. Formation of embryo by the cells other than synergids and egg. Synergids and formation of embryos by the cells other than egg. Formation of embryo by the cells other than egg. Egg in the sense, when male gamete fertilizes the egg, resulting in the formation of diploid zygote. Zygote develops into embryo. So that's what normal development. Here, formation of embryo by the cells other than egg in the sense synergids. What will be synergids? Synergids haploid. So you get only haploid plant body. Example, Asexual, synergids. Antipodals. Antipodals also haploid one. Develop into plant. Example, ulnas. Endosperm normally polyploid only. That condition you get triploid or more than that. So you get Bernophora. Bernophora is a parasite. Last year I studied about it. Okay, formation of embryos by the cells other than in the egg in the sense that don't develop into new plant. They won't give rise to synergies in the sense it will be haploid only. It's not a diploid one, so it won't develop. Antipodals also haploid one. It won't develop into new plant again. But rarely, isn't that rare? It occurs rarely. Formation of embryos by the cells other than egg in the sense it may develop from synergids. Example, Astrolochia. Antipodals. Example, ulmus. Endosperm. Example, Bernophora. But endosperm in the sense it will be triploid or more than that. Development of more than one embryo sac within the same ovule. Normally, within the ovule only one embryo sac develops. But here what happens here? Many embryo sacs are developed. So megaspore mother cell may give rise to two or more megaspore mother cell. Normally, one megaspore mother cell divides by meiosis to form four megaspores. Three degenerate, one becomes functional. That functional megaspore develops into embryo sac or female gametophyte. Here what happens? Megaspore mother cell itself divides by mitosis. Megaspore mother cell divides by mitosis to give two megaspore mother cells or more megaspore mother cells. But all the megaspore mother cells develop into embryo sac. That time you get many embryos. So that's what called development of more than one embryo sac within the same ovule. Then activation of some sporophytic cells of the ovule. Activation of some sporophytic cells of the ovule in the sense maybe nucellus or integument. Some cells of the nucellus or integument give rise to embryo. So they are not diploid only. Nucellus, integuments, they are diploid. diploid only. They develop into embryo. So that kind of embryo, polyembryonic activation of some sporophytic cells of the ovule. So nucellus integument develop into embryos. Example, citrus, syzygium. So what is syzygium? Syzygium, novel word. Citrus, you know very well about that. Lime, lemon, knowledge. Now, casuarina, development of more than one embryo sac within the same ovule, casuarina, souc. Okay. Practical application. Sealing from nucellular tissue in citrus give better clones for orchids. Orchids in the sense planted with fruit trees, areas planted with the fruit trees. Sealing from nucellular tissue, diploid only nucellular tissue in citrus give better clones for, according to better clones, all the plants are same. So better clones for orchids planted with the fruit, places planted with fruit trees. Embryos derived from polyembryony are virus free. Virus free in the sense the plant must be free from virus. Then only you get virus free young plants. Okay. So this is all about polyembryony. What is polyembryony? Occurrence of more than one embryo from single seed, or occurrence of more than one embryo in a single seed, in a seed. It is called polyembryony. Cleavage polyembryony. Then formation of embryos by the cells other than egg. Development of more than one embryo sac within the same ovule. And activation of some sporophytic cells of the ovule. Which cells in the sense nucellus or integument. Then next one about parthenocarpy. Last year we studied about parthenocarpy. What is parthenocarpy? What is a fruit? Fruit is fertilized, ripened ovary is called fruit. After fertilization, ovary develops into fruit. Ovary wall becomes pericarp. Ovules become seeds. So now parthenocarpy in the sense development of fruit without the act of fertilization. The ovary develops into fruit without fertilization. So that process called parthenocarpy. Such fruits are called parthenocarpic fruits. So parthenocarpic fruits are seedless fruits. Parthenocarpic fruits are seedless fruits. Parthenocarpic fruits are seedless fruits. Example, grapes, bananas, papaya, etc. Which classified parthenocarpy into three types. Genetic parthenocarpy. Parthenocarpy due to mutation. Parthenocarpy due to mutation or hybridization. Parthenocarpy due to hybridization or mutation. That kind of parthenocarpy is called genetic parthenocarpy. Example, citrus cucurbita. So hybridization means crossing between closely related plants or unrelated plants. That's what hybrid. But here, closely related plants only. Mutation, any sudden change in the genetic makeup of an organism is called mutation. Environmental parthenocarpy. Parthenocarpy induced by environmental conditions. Environmental changes such as low temperature, high temperature, low temperature, high temperature, then fog, frost, etc. So that kind of parthenocarpy is called environmental parthenocarpy. Chemically induced parthenocarpy. Actually, growth promoting substances like auxins, gibberellins induce polyploidy. Auxins and gibberellins induce parthenocarpy, not polyploidy, parthenocarpy. Auxins and gibberellins induce parthenocarpy. Last year I studied about the seedless fruits are produced by that one. Okay, so that kind of parthenocarpy, chemically induced parthenocarpy. Application of growth promoting substances like auxin, gibberellin induce parthenocarpy. Then significance. Seedless fruits. Seedless fruits have great significance in horticulture. Seedless fruits have great significance in horticulture, and then they have high commercial importance. Commercial importance in the sense, seedless fruits in the sense it will be it will have more flesh than fruits with the seeds. So more edible portion. So the people like to go for that one and then buy even when the price is high. So that's what called, it is commercially important, economically important one. So it has more economical value. So that's what they have great commercial importance. Seedless fruits are useful in the preparation of jams, jellies, sauces and fruit drinks, etc. then it will have more edible portion. So up to this, one lesson is completed for you. The first lesson is completed. Okay, then you will have to write the test. I'll just give you important questions. After that, I'll conduct you test. Okay.